Okay, first a bit of history on the mitochondria. In 1852, Swiss embryologist and histologist Rudolf Albert von Kölliker, another scientist with a great tash, published a book that is reported to contain the first description of the mitochondria. In 1889, Richard Altmann, a German histologist and another scientist with a great tash, observed numerous cytoplasmic granules in many types of cells, and he thought that these were elementary organisms living in the cytoplasm. Altmann called these granules bioblasts. However, it wasn't until 1898 when Carl Benda, with an improved stain, renamed one of the bioblasts the mitochondrium, which is Greek for filament granule. That is, the organelle looked like a filament granule because of the double membrane and the folds in the internal membrane. It looked like the mitochondria contained filaments. Now, at this point, you must think back to an earlier video when we were looking at the sectioning of a bagel and talking about flying horses. Because while all these observations of the mitochondria were correct, they were on fixed, so dead cells, and were in sections. So, some quick facts and figures about the mitochondria in cells. The mitochondria can occupy up to 25% of the cytoplasm, with only the nucleus and vacuoles and cytoplasts taking up more space. The outer membrane of the mitochondria is 50-50 lipid protein and is therefore permeable to molecules up to 10 kilodaltons, whereas the inner membrane is 20-80 lipid protein. We inherit mitochondria from our mothers, roughly 99.99%, with a small number that arrive with the sperm being diluted out by the large number of mitochondria in the oocyte. As you know, the mitochondria is the power plant of the cell. It produces ATP. In fact, it produces 90% of the ATP produced by cells. The mitochondria has an inner and an outer membrane with a membrane space between them. The mitochondria is the site of aerobic metabolism and the oxidation of fatty acids and the metabolism of sugars. Depending upon its energy requirements, a cell can contain 10 to 100,000 mitochondria. However, and this is what you might not know and is not evidence from fixed and sectioned cells, mitochondria form from the adding of new proteins and lipids, and new mitochondria will pinch off from existing mitochondria as required. The mitochondria should not be considered as a single pill-shaped organelle, but as a network of connected organelles. However, it is unclear how the network relates to energy production. But it has now been observed that this network is in contact with the nucleus, the endoplasmic reticulum, the Golgi and the cytoskeleton. The network is dynamic and it shifts between fragmented and tubular forms, with the mitochondria going through rounds of fusion and fission. The mitochondria have their own proteins to drive these fusion and fission events, and the process is regulated by GTP. The mitochondria are not static pills.